Well, good morning, good afternoon, everybody. Welcome back to another exciting Exploring by the Seat of Your Pants. I know we've got some familiar faces in the crowd, but we do have some newbies too. So if you are new to us, we are all about bringing conservation, adventure, and science into classrooms around the world. And you're catching us on a fun day. Number one, it's the second of our triple header today. We've got so many amazing speakers. We just wrapped up with Paul Sokoloff at the Museum of Nature. We're going back to BC a little bit later with Lauren Erling talking about her amazing work. And we are smack dab in the middle with a very cool, totally unplant related broadcast. So you're in for a treat in just a second. I want to note too that we are partnering with this one with the amazing folks at the Gairdner Foundation. So the Gairdner Foundation has joined us for years in showcasing the coolest biomedical and health researchers on planet Earth. Their laureates, the people that they give the Gairdner Award to, have gone on to win Nobel Prizes dozens of times in their history. So they've got a good track record of picking the best and brightest on planet Earth. Now, earlier this week, or late last week, as it were, we were joined by Ed Boyden at MIT talking about his incredible work on neuroscience, and we are keeping the excitement going with the Gairdner folks with Anna Blakney. She has joined us before to talk about her incredible work on RNA, vaccines, the cutting edge of biomedical science out in BC where she is. And she's also, as we were just discussing before going live, an internet personality. She's done incredible work in bringing the magic of her work to the public through incredible TikToks and more. So what this means is that she's a spectacular educator, and you're in for a real treat as we get the chance to learn from her. So without further ado, I'm going to zip my lip and bring in Anna. Thank you so much for joining us today, and welcome back to the program.
Thanks so much, Jesse. Really glad to be here. It's always a treat. So let me just get my screen up.
Beautiful. We are good to go.
Awesome. Well, thanks for tuning in, everyone. And thanks for the really kind introduction, Jesse. It's a real treat to be here. So I'm Anna Blakney. I'm an assistant professor at the University of British Columbia, and my lab is in the Michael Smith Laboratories. And I also teach in the School of Biomedical Engineering. So today I'm going to talk to you about next generation RNA vaccines and therapies. But first, I thought I would give you a little information about me and my career pathway, what my lab works on here at University of British Columbia, and where I think the future of RNA medicine is going. So just to start off, I always think it's really fascinating to learn more about researchers and how they got to where they are. So I'm actually originally from Colorado. That's where I did my undergrad degree, which was in chemical engineering. I then went on to do my PhD in bioengineering at the University of Washington in Seattle, but I also spent 6 months of my PhD at University of Cape Town in South Africa, which, as you can see, is a beautiful place to live. I then did my postdoc in a completely different field than I had done all of my graduate training, so in immunology, and that's when I started working on RNA vaccines, which was at Imperial College London in the UK. And now, obviously, I'm at University of British Columbia in the Michael Smith Labs and School of Biomedical Engineering. So one thing I wanted to point out before we dig into the science is one of the reasons I think science or engineering or really being in academia is such a great career is that it's really like a passport. So growing up in Colorado, which I still think is a great state and a great place to live, I always thought that I would end up there. But through research, I've had the opportunities to travel and live in many amazing cities, including Boston, Pittsburgh, Cape Town, Seattle, London, and Vancouver. And so I always, I think this gets a lot of younger people and trainees excited because you may not realize it, but it's one of the easiest jobs to move around and see different research labs in different countries. So if that's something that excites you, know that this could be a good fit. So the way that I got into the field that I work in now, which I would call immunoengineering, is, as I said, I come from a very fundamental chemical engineering training. And when most people think of chemical engineering, they think of kind of this diagram that I have here, which is kind of large scale, industrial processes. And historically, this was always for oil and gas applications. So many of my colleagues and fellow students went on to work on oil rigs and what I would say is quote unquote real chemical engineering. But because of my training in immunology, so this is the study of our immune system, and I was always really fascinated with this and how we could train our immune system to help treat and prevent disease. this is how I arrived at this idea of immunoengineering. So what this means for those of you who aren't familiar with this term or this field is that we engineer the immune system. So in the ways that engineers would design a system to have a predicted output, we try to design our immune system to act in a specific way. And I'll talk about this a little bit more in the context of our work with RNA vaccines and therapies. So there's kind of three different things that my lab works on, or three pillars of the lab. In the past few years, obviously the field of RNA vaccines has changed a lot since I started working in this field in 2016. But I like to say now the plane is off the ground. So we've shown that these RNA vaccines can work pretty well against a viral pathogen, COVID-19. But I think there's still so many things we can improve. And so that's what my lab is really interested in doing, is seeing what can we do next and how can we make this technology even better. So the first thing we work on is RNA molecular engineering. And what this boils down to is how can we engineer this RNA to have a longer half-life, so to last longer in a patient or express more protein within a patient. We also work on what's called biomaterials design. So the RNA has to be packaged in tiny nanoparticles to get into our cells. And a lot of this technology was actually pioneered here at UBC by a Gairdner Award winner, Peter Cullis, using what's pictured here, which is lipid nanoparticle technology. So what we're trying to do is to get the RNA to specifically the cells and tissues where it needs to go. And then we're really interested in what I call like the frontier of RNA medicine. So we're interested in making better vaccines, but also using RNA for other applications. And I'll talk about what all those other applications are in a bit. So one of the questions I often get asked is, why do we even need new types of vaccines, right? We have lots of vaccines out there that are approved and many of which work quite well, right? So some of the motivation for us for working on this is that there's still a lack of efficacious treatment for many devastating infections. So just for example, HIV-1, we've been trying to make a vaccine for that for over 30 years now and has eluded us this whole time. We're also interested in using vaccines to confront the emergence of multi-drug resistant bacteria. We're always looking to improve the safety profile of licensed vaccines. So how can we make them even better and more tolerable for patients? One that I think we can all relate to now is disease outbreaks. So sometimes a new pathogen like SARS-CoV-2 arises, and we need to be able to make a vaccine rapidly in response to this pathogen. We are also need to deal with kind of the changing age structure of the population. So as people live longer, this means we have more elderly people. And as you age, your immune system doesn't respond as well to kind of traditional vaccines. And so we need to think about different strategies for this. And then, of course, we're always looking to reduce the cost and time required to produce vaccines. And this is one of the motivators for our work as well. So some of the applications that we're working on is specifically looking at RNA vaccines for bacteria, RNA vaccines for disease outbreaks, and then reducing the cost and time required to produce vaccines. So you may be wondering, how do you even make an RNA vaccine against a new pathogen? So this is a very simplified version of the process, but we can do this for basically any pathogen where we know the protein that we want to encode. So this is a schematic of the SARS-CoV-2 virus, and many of you may be aware now that it has a protein located on the surface of it that's called the spike protein. So we take the code for that spike protein and we put it into our RNA. We then put that RNA into the nanoparticles that I alluded to earlier, and that's our vaccine. And then we start our preclinical animal studies, usually starting in mice. So one of the things that was really remarkable about the RNA vaccines was the initial safety and efficacy of them against COVID-19. So this is some of the data from the Pfizer and BioNTech vaccine showing in two different curves. So the blue is the placebo. So this is the group of patients that didn't receive a vaccine. And then in the orange, this is the group of patients that did receive the Pfizer and BioNTech vaccine. And they always do the statistics on here. That's what's down here with the 95% confidence interval. But I think what I'd like to see is data where you don't need the statistics, and you can very visibly see this difference between the incidence, so how common it was to get the infection, between the placebo and the vaccinated group. So initially, the 2-dose regimen of the Pfizer and BioNTech vaccine conferred a 95% protection against COVID-19. And then the safety profile over a median of 2 months was similar to that of other viral vaccines. And this was why the Pfizer and BioNTech vaccine was approved. It was really outstanding results for this initial vaccine. So where do we go from here, and what are the challenges with vaccines? So one of the priority areas in the field is really minimizing the dose of RNA, which we know is really highly correlated to the side effects. So this is some clinical trial data from one of Moderna's early phase 1 clinical trials. So they're one of the makers of one of the mRNA COVID vaccines. And I'll walk you through what they've done here. So in this phase 1 trial, they were mainly looking at safety, or kind of the side effects that they were seeing in patients. And they used 3 different doses of the vaccine. So 25, 100, and 250 micrograms of RNA. And they've looked at all these different side effects, and they've categorized the patient's response as mild, moderate, and severe. And again, I think this is very easy to visualize data where that you see that by increasing the dose, you increase both the frequency and the severity of side effects. So what this means for the field going forward is that it's really a priority to minimize the dose of RNA that we're using for these vaccines and therapies, especially for non-pandemic contexts, such as people receiving an annual flu vaccine or similar, and even more important for patients that may be receiving therapeutics even more frequently. So if a patient has to receive a therapeutic every week or every 2 weeks, they really don't want to have some nastier side effects that we might have seen from the initial RNA vaccines. So this is one of the things that my lab works on with a type of RNA called self-amplifying RNA. So if you've never heard of this before, it's just a type of messenger RNA, and I'll walk you through how it's different. But the main takeaway here that you need to understand is that using self-amplifying RNA enables a lower dose of RNA so that we're able to dose to the patient less and potentially have less side effects than a typical messenger RNA. So on the right, we have a typical mRNA. This is a picture of the cell. So once an mRNA gets into a cell, it engages with the ribosome and you get translation of your protein. This is part of the central dogma of biology that you may have learned about. And basically, instead of manufacturing the protein ourselves, we're using the patient to manufacture the protein in their own cells. So self-amplifying RNA starts out very similarly. Once we get a copy of saRNA in a cell, it engages with the ribosome just like mRNA does. You do get a little bit of translation of your protein antigen, so that's for your vaccine, but more importantly, you get translation of a different type of protein called a replicase enzyme. And what this replicase enzyme does is it engages with this initial strand of RNA and makes many copies of it. So exact copies of it, it's acting as like a photocopying machine. And I have pictured just 3 copies of the saRNA here, and that was for my sanity in BioRenner, but it's actually thousands of copies of RNA within the initial cell. So because of that, you get so much more protein that's translated because you have many more copies of this template here. So because of this, we're able to use about 100 times lower dose of a self-amplifying RNA than a typical messenger RNA. So this was the technology that I've been working on since 2016. And obviously in early 2020, the COVID-19 pandemic started. And so I was actually part of a team at Imperial College that worked on an saRNA vaccine that went into a phase 1 and 2 clinical trial. And so I wanted to tell you a little bit about that process today so you can understand more about how we develop these RNA vaccines. So first, what I think is most impressive and so cool about making these vaccines is just the timeline of actually making and manufacturing and developing new RNA vaccines. So I'll walk you through our timeline and then tell you how this fits into the context of the field today. So the circulating strain of SARS-CoV-2 was made publicly available on the 10th of January, 2020. Our team at Imperial College London accessed the sequence on the 20th of January. I then coincidentally flew into Vancouver to interview for my current position at the Michael Smith Labs on the 21st of January. So as you can imagine, this was a very busy time. We ordered the DNA that we need to make the vaccines on the 22nd of January. We then assembled them into the plasmid, also needed to make the RNA on the 28th of January. We made the RNA and confirmed that it was expressing the right protein on the 12th of February. and then started our preclinical immunogenicity studies on the 13th of February. We started our rat toxicology studies on the 30th of March. So before you go into any human, you have to do a rat toxicology study. And so that's what we started. We submitted our paper on this on the 22nd of April, and then started our human clinical trials on the 19th of June. And so we went from our team of, just a small group of scientists of making a new vaccine starting on the 20th of January to putting it into a human clinical trial on the 19th of June. And so this, I think, really, shows us how powerful this technology is that as long as you know the protein that you want to make, you can make any new RNA vaccine in a very reasonable amount of time. So it's been really exciting to watch the field develop as well. And I figured some of you may not know this, but there's actually 2 approved saRNA vaccines now. So the first is developed by a company in Seattle called HDT Bio, which is approved against COVID-19 in India. And then more recently, an saRNA vaccine also against COVID-19 was developed by Arcturus Therapeutics, which is based out of San Diego, is approved in Japan. And so I know they've partnered with a company in Australia called CSL Seqirus, and are looking at entering other markets. And so I think we'll start to see saRNA vaccines against COVID available more places in the world starting soon, which is really exciting. So how do we make vaccines, and why is RNA vaccine such a more scalable technology? So for those of you who aren't familiar, kind of the historical way of making vaccines was, I think you can notice some of this is touching on my chemical engineering roots, this large scale equipment and large processes. But actually on the left here, this picture with the eggs is how we made the flu vaccine in the year 1957. And actually that's still how we make the flu vaccine today. So as you can imagine, this is a very time and resource intensive process. The virus is grown up in eggs and then further processed to make the flu vaccine every year. And so because of this, it means that we have to start making the flu vaccine very early so that it's ready in time for flu season. But that means we decide in February of each year what the strains are going to be. But in that time, the flu vaccine just continues to mutate and change. So if we can cut down on that time to make it, then it will probably be a better flu vaccine. So many other types of vaccines are made in these kind of large scale bioreactors where we grow cells, and the cells are the ones that make the protein or the virus that we then have to downstream purify and further formulate. So what's fantastic about RNA vaccines is that they're completely synthetic. So we don't need any cells to grow and make these, and you do it the same for every single RNA vaccine that you're going to make. And the scale is much, much smaller. So this is a scale of about 100 mils, which is about the size of a coffee cup you might drink. And we can do this at the benchtop, and in 100 milliliters, you can make 1 million doses. So even in my lab here, we could easily make 1 million doses of an mRNA vaccine, which is pretty outstanding. So actually what we encounter most of the time in the field is that oftentimes the equipment manufacturers don't make equipment small enough for the needs that we need because they're used to making these huge bioreactors for other types of vaccines. So just to put this into perspective and how powerful this is I like to visualize by thinking about how many Olympic-sized swimming pools would it take to vaccinate the entire world with an RNA vaccine. So I did this calculation for the Pfizer and BioNTech vaccine specifically. And there's about 7.7 billion people in the world. Everybody needs 2 doses initially of the vaccine, which is a total of 15.4 billion doses total. Each dose is a volume of 0.3 milliliters. So that gives us a total required volume of 4.62 million liters. Each Olympic-sized swimming pool is about 2.5 million liters. And so you just need 2 Olympic-sized swimming pools to vaccinate the entire world with an RNA vaccine. Now, this might sound like a big volume, but it's also a very doable volume, as evidenced really by the pandemic when we made, billions of doses and billions of people around the world have gotten RNA vaccines. So this is really what makes this technology so scalable compared to the traditional way that we make vaccines. So, in the context of the pandemic, one thing that was really difficult was that there actually wasn't a ton of infrastructure to scale up and make these RNA vaccines. So for the vaccine that we brought into clinical trials, it was really a global experience at the time. So we got our RNA from a company in California. We got our lipids from a company called Acuitas, actually here in Vancouver, just a few blocks away from where I am now. They were shipped to a company in Austria that made our lipid nanoparticle formulations. Those formulations were then shipped to Italy to be put into vials, and then all of that was shipped back to the UK to just start our clinical trial. So this is something that many countries are working on changing now, and we actually most recently got funding from the Canadian government to start a full GMP manufacturing facility right here on UBC's campus. So we'll have the ability to do this locally soon. And I think that will continue to be a trend worldwide. So we no longer need these like big centralized vaccine manufacturing centers. And instead, you could imagine having some of these more locally where you could produce, your seasonal COVID and flu vaccines, but also personalized cancer vaccines or other types of applications that you might be using RNA for. So I present this slide in almost every presentation that I give, and it is the most difficult slide to keep updated because there's so much momentum in the field. But what I'm trying to get across is the RNA vaccines or approved RNA medicines that we have today, what's currently in clinical trials, and where this may be going in the future. So today we have a number of approved mRNA vaccines. So probably most of you have heard about the approved Pfizer and BioNTech and Moderna vaccines, and also the approved saRNA vaccine from Arcturus and CSL. There's also now, those are all for COVID-19. There's also now an approved mRNA vaccine against RSV, which Moderna developed. And then there's so many mRNA vaccine and therapeutic clinical trials going on. Most of these are in the infectious disease and oncology space. So for some infectious diseases like influenza, tuberculosis, malaria, rabies, a whole range of diseases. And in the oncology space, again, it's a whole range. So HPV, melanoma, pancreatic cancer, systemic immunotherapies. It's very wide ranging, which is exciting to see. And some really exciting results from both some recent melanoma and pancreatic cancer trials. And in the future, that you could use this platform for so many different things. So you could use this for rare diseases where, just a protein is being made incorrectly or isn't being made at all. There's been a lot of momentum in the field for personalized cancer vaccines. So tailoring this to each patient, autoimmune diseases, cardiovascular diseases, there's really a variety of applications. So it's really been interesting and exciting to see the field take off and start pursuing these different applications that hopefully have a big impact on human health. So one of the things, and I want to wrap up this presentation just by talking about how important I think science communication is and our work as kind of mobilizing knowledge as scientists. So this may seem like a simple concept, but if nobody wants to take the vaccine, there's no point in making it. But I think this is actually very hard for some scientists to grasp. Like you see, they're so excited about the technology. They've spent so much time developing it. But this was a study that was done by Heidi Larson, who runs the Vaccine Confidence Project in London, that was published in 2020. And basically, they did this study all over the world and asked people to rate whether they thought vaccines are safe, vaccines are important, and vaccines are effective. And so the more blue it is, the more that people in those countries agreed with those statements. So you'll notice that pretty much worldwide, people leaned heavily saying that vaccines are important. But there's a big disparity between thinking vaccines are important and thinking vaccines are safe and effective. And so this really gets us thinking, like, where does vaccine hesitancy stem from? And what is this missing link between thinking that vaccines are important but not safe or effective? And so this is why I think public engagement is so important. So what I've observed is that the pace of science is not matched by the pace of public communication. So we saw this very evidently, in January 2020, there were still a lot of scientists who didn't know what an RNA vaccine was. And now billions of people have gotten them just, 4 years later. And so because of this, I think we just need to do better about shortening that gap between, the progress that we're making in our labs and telling people about that progress. So one of the ways that I've done this is through TikTok, which many people think of as a platform for dancing teenagers. I started doing it in October 2020 as part of an organization called Team Halo. So this was a collaboration between the United Nations and the Vaccine Confidence Project. And what I found is that it's really an elegant platform to show people how we make and test vaccines and directly confront misinformation, as well as just answer people's questions that they may have about RNA vaccines. And part of the reason I think it works so well is that the videos are short and really engaging and offer a palatable amount of information. So instead of having like 20 minutes like I have today, you have like 15 seconds or 30 seconds to really get a concept across. So one of the most enjoyable parts of this for me is really recognizing the scientists behind the COVID-19 vaccines. So there are two pictured here, but I've had a number on my TikTok. So this is Bob Langer, who's a professor at MIT. He was one of the co-founders of Moderna. And this is Peter Cullis, who's a professor here at UBC, who started Acuitas, which is the company that formulated the Pfizer and BioNTech vaccine. And what's really heartwarming about these videos is that you can see in the comments, there's all of this positive feedback, like, thank you for saving the world, real life superhero. I would love to get it. Thank you for saving lives. So grateful for you. And what this made me realize is that people really appreciate scientists, but they also have no idea who they are. Like, if you talk to anybody in my field, both Bob Langer and Peter Cullis are like idols. And it's really funny that like the average person doesn't know who they are. And so part of my mission is to make more scientists household names. So in conclusion, kind of summarizing everything we've seen today, we saw that the mRNA vaccines have enabled a rapid response to the SARS-CoV-2 pandemic. On the manufacturing side, mRNA vaccines are made synthetically, and so they require a much smaller footprint and time frame to manufacture than traditional vaccines. We've seen that the side effects for RNA vaccines are proportional to the dose of RNA. So this is a priority to minimize the dose of RNA in our work. And last but not least, communicating to the public about science is imperative. So I think this is important, not just for RNA vaccines, but for any sort of science that we're doing, which is why I love doing things like being here with Jesse and you all today. So with that, I'd just like to thank all the members of my lab and funders that have contributed to this work, and happy to take any questions.
Outstanding, Anna. So much fun to get to feature you on. And I mean, I followed the pandemic, obviously, assiduously over the course of the several years that we were all living in it and locked down and everything else. But it's so amazing to get to highlight the story of vaccine development, how important it is, how much it's changed in the last years. Like, this is the golden age of biomedicine in this field. So lucky you to get to be involved in both sharing that story and actively doing amazing research in it. It must be so much fun. We've got a bevy of questions already, so we're going to dive right in with those. The first, no pressure, what do you find to be the biggest advancement in vaccine medicine over all time? No pressure.
Over all time. Oh, that's a really fun question, and fun to think about just how much the field has changed. So what I think is the biggest advancement, or I think what it will enable, is this idea of personalized medicine. So we've started doing this a bit more for like the personalized cancer vaccine. So if you're not familiar with how this works, basically if a patient has a tumor, we're able to take a small biopsy of that tumor, pull it out, sequence it with all of the technology that's available now, and know exactly what are the proteins that are being expressed on that patient's cancer cells. So we can then go through and say, okay, these 20 proteins we think are a good target for the immune system, make an RNA vaccine, and then you have an RNA vaccine that's perfectly tailored to your cancer. And so that's the fact that like all of those things line up and are feasible, I think is, really impressive. But this is not necessarily just useful for cancer. I think this could also be very impactful for like rare genetic diseases or metabolic disorders. And the difference for this is it still needs to be perfectly tailored to one person. But for cancers, there's probably some more common mutations that we understand. For these like rare genetic diseases, it's much less well understood. So I think there's a lot of development that needs to go into it. But as soon as you know like what you need to encode, you can very easily make an RNA medicine for that.
Which is amazing. I want to harp on the cancer thing as a personal question. So you get this protein that you've taken a biopsy from the tumor. I mean, the thing with cancers has always been that it's your own cells run amok. So how do we know that the proteins that we're getting from that biopsy aren't found in other parts of the body that we know? Is it because we have such a good knowledge of the body outside of cancers, or what?
Yeah, I would say that's actually probably the hardest part. So we have a better idea now. I guess there's like two parts of it. One is that protein on other parts or on other cells or tissues in your body. We have a pretty good idea of that now because people have done like proteomic mapping of like all different types of cells. And so we have kind of a sense of where things are expressed. But the harder part is knowing, okay, is this protein going to give you like the right immune response to that tumor? And so now, again, that's actually where people are using like machine learning and AI to understand, okay, if we use these, here is like the outcome that we see with the immune system. And then it's an iterative process to know. So with each patient, hopefully it's getting better and better to design better personalized cancer vaccines.
Very cool. Thank you for that. And for looping in AI. We've got AI, mRNA, TikTok. All we need is like lasers and quantum, and we've covered like all the bases of cutting edge.
Almost there.
We got a cool question, and how you want to take this is up to you. So what are the types of vaccines? Do they have specific categories? You're talking about different diseases that they're for and how they're designed, but is there any sort of like, this is this kind of vaccine versus another that you can share with us?
Yeah. Yeah. So I didn't include a slide on this today, but that's a good question and an interesting observation, because if you think prior to the pandemic, probably nobody knew who made the vaccines they're getting or even what type of vaccine they were. So this is like a kind of just funny aspect of society, but people learned a lot about RNA vaccines during the pandemic for the most part, which I'm very grateful for. So yeah, there's a variety of types of vaccines. So I guess maybe I'll start from a high level of what we're trying to do with the vaccine. So with any vaccine, we're trying to train your immune system to recognize a protein that's found on a pathogen, a virus, bacteria, etc. So there's many ways of introducing that protein into your body. So we could just introduce the whole virus itself. Usually we attenuate it or kill it in some way, so it's not actually actively infecting you for a vaccine. But we can also just manufacture that protein itself. So either we take some sort of virus and inactivate it, or we identify the protein and then just manufacture that protein itself. So those are kind of the traditional ways we made vaccines. There's some other ones. So sometimes with bacteria, we'll use some of the glycan groups from that bacteria or polysaccharides. That seems to work well for some vaccines. But basically, historically, it was just taking some sort of pathogen and making it so it wouldn't infect you, and that was the vaccine.
The origin story of sort of vaccinology, is that the phrase? Good. It's so weird. I've never said it out loud before. With smallpox and cowpox and sort of the how vaccines got their start is one of the coolest stories in the history of science. As a field, it's so neat, and that's why Anna's so happy to talk about it all the time.
Yeah, totally. Actually, I have a funny story about that. So, Jesse, do you know, or I guess does anybody know who the original maker of vaccines was, or who's the first person to have who was credited with making the first vaccine?
Jenner, yes?
That's right. Okay, so the story was he realized that, so this was the smallpox vaccine, and he realized that the women who worked with cows, which were called milkmaids, were immune to smallpox because they were getting a version of smallpox that infected cows, the cowpox, which didn't kill them, but made them immune to smallpox. And so he decided to do this experiment to see whether this actually worked and if his hypothesis was correct. And so he tested, it's very different from how we test vaccines now, but he tested it on his family, himself, other people, and found that it worked. And that was how they developed the first vaccine. And I have a very cool kind of personal connection. I don't know if you can see it behind me. Oh, it's kind of, so behind that plant, you can't see very well, but I used to live in London, and one of the people who I played tennis with has a collection of what are called book plates, which is a piece of paper you used to put in your book. And when I was leaving, they gave me Edward Jenner's book plate, which I can show you.
Oh, save the plant. This is so cool for a nerd like me that you have this. Like, this is really awesome. I would want this too.
It's extremely nerdy. So this was Edward Jenner's actual book plate that he put in his books, which is now in my office and serves as a daily inspiration to do our work.
I think the Edward Jenner story, like with cowpox, is the most, like, it's the easiest way to explain the origins of vaccines and why they're so important, because it's like there's this disease that wiped out millions and millions and millions of people for centuries, and we found an easy way of giving people something that would not kill them and not seriously injure them. And it was before, some of the challenges that people have with vaccines. We're not going to get into them all now because we want to highlight the good parts of vaccines. But these are incredibly safe and effective technologies. And if you start with the origin story, I think that really drives the point home for people that would otherwise might be a little bit skeptical about them.
Yeah. And I, for one, am glad that we no longer get vaccines where you take pus from a cow and use that to inoculate people.
I would much rather have a shot than cow pus. That's our unexpected takeaway for the day. We might have that as the byline for the next time we do this broadcast. No more cow pus, a future of vaccines. Anna, we've got some more questions before we wrap up. Time flies and you're having fun, so we've got time for about 2 more before we finish today. We've got one. What are some struggles you experience when working with vaccine medicine? So in your job, is there a big challenge? Is it I don't know. Tell us more.
Yeah, totally. So it's not always rainbows and unicorns. Actually, a lot of science is failure, right? So actually, one of the biggest, most challenging kind of like phases of my career, I would say, is actually when I started working on RNA vaccines. So the lab that I was working in, like nobody had ever made RNA or an RNA vaccine before. And so I spent probably at least a year trying to make RNA and to actually get it to work, and nothing was working in that time. And so that's a really long time for your experiments to not be working. And so, yeah, because of that, it was like a ton of troubleshooting, really understanding the system. But looking back, it's something I'm really grateful for, because then when I went to start my own lab here at UBC, I had been through all of the possible troubleshooting that you could do. And I knew how to set everything up in a very efficient way and get it to work initially. So yeah, there's definitely been some really tough times. But I think as a scientist, it makes you have to learn how to be resilient and get through the failures. And every failure is actually learning, right? We take something away from it, but it doesn't always feel great.
I'm so glad you mentioned being a failed experimentalist, because almost no one's ever mentioned that in the history of the program. And so when I did science in university, I'm a biologist by background, like we'd go in and there'd be like 50 students and we'd have like a DNA assay to do, or we'd have to take something out of a fruit fly. And I was the worst at all of those. Like, I am horrendously bad at them. And I mean, I'm sure if I'd kept trying, I would have gotten better and better and everything else. But like, it was a little demoralizing to go into the lab and everyone's like, oh yes, my experiment worked perfectly like that. And here I am being like, I crushed the fly again. I crushed the fly again. Oh no, crying softly to myself in the corner. So thank you for that. And I honestly, I'd love to highlight for students that like, it's not all rainbows and unicorns. So much of getting money or grants for the work that you do is a huge challenge, and you're often rejected. Finding the right lab. You really liked hopping around and going to these various amazing places. Some people find that a tremendous challenge. So it really depends on your outlook and approach. But there's always going to be a difficulty in whatever job you do, and the goal is to find as many fantastic things about it and the way to sort of transcend those things that make it worthwhile. So thank you for that, Anna, very, very much. Do stick around with me after we're done live. But as our final question while we are live together with all our students, is there any final message you'd like to share about your lab, about the future of vaccinology, about any of this that we want to leave off with all of our kids today before we say farewell?
Yeah, sure. So, I think the field of RNA medicine is just starting, really. We have a few approved vaccines and other types of therapies now, but I think the future is so bright. And so maybe what I'll leave you with is kind of my outlook on this, but I think it's helpful for everybody actually, which is, it's like partially written on my board behind me, which you can't really see, but it says, what would you do if you weren't afraid? So I think right now the challenge for us in the field is thinking about, okay, what can we use this for that may be very impactful and maybe very challenging right now? It could never work, but it's really taking those risks with the potential that, this may be an amazing vaccine or therapeutic one day. And so this, of course, applies to our work, but also many of you in your future careers. What would you do if you weren't afraid?
Anna, there is no better message than that. Thank you so much for joining us again. And I'm going to take that into my own work as I go forward. So I appreciate your expertise, passion, all of it. I can't stress enough for our students. Check out Gairdner Awards, and specifically check out Anna on TikTok. She is incredible, and there's way more than we could possibly cover in one broadcast, but she is a spectacular advocate for science, technology, engineering, and more. And with that, Anna, thank you so much, and have a wonderful rest of your day. We're end live for now.